Good afternoon, everyone. I'm Kata Kóta from the Institute of Experimental Medicine. This is a brain research center. And today I would like to introduce you our project about brain research. We examined the brain regions that can innervate the paraventricular thalamic nucleus using genetically modified mice and viruses. Well, at first this may sound a bit complicated, but I hope I will be able to explain it to you. But first, you may wonder, why is this important at all? So let's start at the beginning. Of course, surely you feel occasionally stressed, or it happened to you that you couldn't sleep well before an exam or a test because of extreme alertness. Of course, these happen to everyone, but in serious cases, this can cause serious diseases. For example, an acute stressful event can lead to acute or post-traumatic stress disorder later, and chronically persisting stress leads to anxiety syndromes or depression, or it can cause any other cognitive dysfunctions. And extreme alertness is a common cause of insomnia, which is a sleeping disorder. And of course, these two also influence each other. And there's a thalamic region called the paraventricular thalamic nucleus that was indicated to take part in the development of both types of these diseases. So first, I will talk about the thalamus a bit. This is the human brain, and here you can see the thalamus. And it consists of units called nuclei. And surely you have learned that the thalamus transfers the sensory information from sensory organs like the retina to the cerebral cortex. But, and this is true, but actually it's not just the sensory information, but every other kind of information from the brainstem and the hypothalamus that must pass through the thalamus before they reach the cortex. So these thalamic nuclei play a key role in not just sensory, but also in various motor and cognitive functions. For example, this is a human study where the researchers found that this upper middle portion of the thalamus is involved in hyperarousal, so in extreme alertness associated insomnia. And more specifically, in this upper middle region, this is the paraventricular thalamic nucleus that can take part in these phenomena. Here you see the human brain compared to a mouse brain, because we use mice for the experiment, and this red part shows the location of the PVT in the mouse brain. And the PVT cells contain a protein called carotinin, and if we mark this protein, we can see the borders of the PVT. As I mentioned, the PVT is involved in the regulation of wakefulness levels. And on this video, you see a sleeping mice, and there's an optic fiber implanted into its brain. And our research group activated the cells of the PVT, which is indicated by this shine. We used light to activate the cells. And as you saw, the mouse immediately woke up from the sleep because of the activation, and it started to move around actively. And our research group also found that the PVT is involved in the regulation of behavioral and neuronal stress responses. An acute stressful event can cause serious behavioral changes in mice. And when we externally inhibited the PVT, we could prevent these changes caused by the stress exposure. And the PVT also takes part in motivated behaviors, feeding, and drug-seeking mechanisms. So as you see, externally manipulating the PVT activity leads to serious changes. So it is very important to know that which brain areas are able to increase and decrease the PVT activity in natural conditions. In the brain, you can find some excitatory neurons and inhibitory neurons that can increase and decrease the activity of other neurons. So our research aim was to find out which brain areas contain excitatory and inhibitory neurons that innervate the PVT. And for this, we had 3 conditions to fulfill. Firstly, we have to able to selectively label excitatory and inhibitory neurons in the brain. Then we also have to see these neurons. And finally, we have to find out from which brain regions the axons come to the PVT. So to answer the first question, we must take a look at the Cre-log space system. The Cre recombinase is a special protein. It's an enzyme which can recognize certain sequences in the DNA. These are called log space sequences. And when the enzyme recognizes these sites, it can cut the DNA and modify it in different manners depending on the orientation of these log space sites. So, for example, if there is a gene between 2 log space sites, then the Cre enzyme will cut this gene out. But if the gene between the log space sites is so in the first case, the log space sites was in the same orientation, but in the second case, they are opposed to each other, and in this case, the gene is not cut out, but its orientation is reversed. And it can also happen that these log space sites are on different DNA molecules, and in this case, as you see, these DNA segments exchange their positions. But for us, the case of the inversion will be important because for the experiments, we used genetically modified mice which contain this Cre recombinase, but which is very important that only the excitatory or only the inhibitory neurons contain this enzyme, so we can manipulate them selectively. And after we inject viruses into specific nuclei of the brain, and then they can infect the neurons. And these viruses are Cre-dependent because they contain the genes of fluorescent proteins. But as you see, in neurons without the Cre enzyme, these are in the wrong direction, so they cannot be expressed. And there will be no protein production. However, in neurons that contain the Cre enzyme, the gene is inverted, so there will be a fluorescent protein, and then we can see these neurons. So this way we can selectively label excitatory and inhibitory neurons in the brain. Then our second question is to see what we have labeled and also other markers in the brain. So we prepared the brain slices and used a technique called immunostaining. And the principle of it is the specific binding of antigens and antibodies. As you know, antigens are every foreign molecule for an organism, and the immune system can produce antibodies which specifically bind these antigens, but only one kind of antigen. And we can use this to our advantage if we attach a fluorescent dye to these antibodies, because then we will be able to see fluorescence at the site of the protein we want to label. And in our case, the antigens are not bacteria or any other pathogens, but firstly, they can be the viral fluorescent proteins. This is to make their signal even stronger. Or it can be any other marker molecules in the brain, because of course we want to see where we are on brain slices. So for example, we can mark the carotene protein to see the PVT. And okay, so now we have labeled the excitatory and inhibitory neurons, and now we can also see them. But how does this answer the question from where the axons of the labeled neurons come to the PVT? And the answer is that these viruses won't stay in the place where we injected them, but they can travel along the axons of the neurons. And there are more types of viruses that we've used. The first is the retrograde virus. And if we inject a retrograde virus in the brain, then the axon terminals will take it up, and it travels backwards to the cell bodies, so we can identify them. Here you see an example. This is the virus injection in the PVT, where the axons have taken up the virus. And this is another brain area in the brainstem, and these pink dots are the back-labeled neurons. So this way, we could identify many brain regions that innervate the PVT. This is an example in mice where the excitatory neurons are labeled, but we also did these experiments in the other case in mice where the inhibitory ones are labeled. And the other virus that we've used is called anterograde virus, and it's the opposite of the retrograde ones, because now at the injection site, the cell bodies will take up the virus, and then it can travel forward to the axon terminals. So this way we can examine the axons. Here's again an example, in this case in mice with inhibitory neurons labeled. This is a hypothalamic nucleus where the injection site is. And here you can see the axons in the PVT, especially in the 60-time-fold enlargement microscopic image. The blue ones are the axons around the yellow PVT cells. So to summarize, it was known that the PVT can regulate various functions which potentially participate in the development of diseases associated with extreme alertness or stress. But it was unknown that which areas can increase or decrease the PVT activity in natural conditions. So we used Cre-containing genetically modified mice and Cre-dependent viruses. And this way we could selectively label excitatory and inhibitory neurons in the brain. And we used retrograde viruses, which go from the axons to the cell bodies, and we mapped the brain areas that innervate the PVT. And we also used anterograde viruses, and we injected them into PVT innervating areas, and then we examined the axonal pattern in the PVT. As a conclusion, I could say that with the help of genetically modified mice and special viruses, we can map the anatomical connections between various brain regions, which could help understand how a nucleus works and how they participate in different mechanisms. I would like to thank our research group, the Thalamus Research Group, their precious help, and thank you for the attention.
Thank you for the nice presentation, and now please take your certificate. Kata Kóta. Next speaker is Dorina Kovács. She is from University of Szeged. She is at 5th year of Faculty of Dentistry. The scientific partner institution is Hunren Biological Research Center. Mentor is Bálint Kincses, and the title of lecture is Better Together, In Vitro Efficacy of Beta-lactam Antibiotic and Inhibitor Combinations.
Thank you very much. Ladies and gentlemen, dear students, teachers, I'm really grateful to be here and present you today my work. I'm going to talk about antibiotic resistance and beta-lactam antibiotic and beta-lactamase inhibitor combinations and why they are actually better together. So, I think that we all heard this phrase before, maybe from our teachers, parents, or even from the song. If you ask me, it isn't really much help in a difficult situation, but I'm going to show you why this is actually really true in the case of bacteria. So an infection is mostly, it's mostly caused by a bunch of bacteria, and if we want to treat it with antibiotics, then the majority of the bacteria are going to be killed. But a small amount of bacteria can be left after the treatment, and they want to fight. They want to be able to defend themselves in the future against the antibiotics, so they might go through a mutation, and after they spread again, and we want to use the antibiotics again against these bacteria, then they survive, and so we can say that they are resistant to the antibiotic. You may ask why a small amount of bacteria is left here. Well, there are a bunch of reasons. I would point out one. When you get an antibiotic prescribed, or patient gets an antibiotic prescribed, they start to take it, and after they feel better, they say, okay, I don't need the antibiotic anymore, I just won't take the drugs till the end, and thereby they leave the antibiotics in their system, so there is time for the mutation and place. So take-home message is always take your antibiotics until the end to be able to prevent this from happening. There are a lot of different mechanisms of antibiotic resistance. One of them are different enzymes, and we are focusing on beta-lactamases. These are a type of enzymes that are able to hydrolyze beta-lactam antibiotics, and thereby they are providing resistance to the bacteria. But in the case, if we inhibit this process with beta-lactamase inhibitors and we combine them with beta-lactam antibiotics, then the resistance might be gone. So when combining these two or different antibiotics with inhibitors, we might have actually effective antibiotics again. So a bit about our project. Our aims are to identify which resistance genes, mostly which beta-lactamases, are prone to cause resistance at the highest rate. Then, of course, we also want to determine which are the most dangerous strains spreading in our environment. And of course, we want to be able to say which are the most effective combinations against the strains that are containing the genes. So these are all connected to each other at the end. About our project, well, in the lab, the project that is focusing on beta-lactam antibiotic and beta-lactamase inhibitor combinations has two different parts, and I'm going to talk about my part, the strain level measurements. So basically, I have a strain collection of nearly 230 different strains. They are partly Klebsiella pneumoniae and Acinetobacter baumannii. When creating this strain collection, my goal was to have the highest genetic diversity possible, so to to get as many different genes, beta-lactamases, as possible. And since we know the genetic background of every strain, because they are all whole genome sequenced, I was able to pick out those that I want to use in the experiments. I conducted MIC measurements with these strains. The MIC is the minimum inhibitory concentration. You might have heard about it, but just a refreshment. So the MIC is the lowest concentration of an antibiotic that is able to kill the bacteria. And I'm using such 96 level plates that is shown on the slide, meaning that I've got different strains in every single well. And thereby, and then I add the antibiotics, and thereby I'm able to tell what is the MIC level, and then after that, whether the strains are resistant or susceptible to the particular antibiotic. I brought you a table as well about the combinations that we are working with. So you can see in the first column the beta-lactam antibiotics, then the beta-lactamase inhibitors, and I would point out the column of the clinical phase, since in our experiments, we are mostly focusing on combinations that are in some stage of clinical development, because we want to be able to say or tell predictions about whether the combinations are going to be effective in the future when we actually start to use them worldwide in the clinics. But we also have a few old combinations, the ones that are completed the clinical trials. These are kind of controls for our measurements. And on the two other columns at the end, I showed you against which strains, which species I tested the different combinations, because not all inhibitors are, good against every species. This is actually data from the World Health Organization or other publications, but which combination is great, or even which combinations are good to test against the species. So now let's move on to the results themselves. I won't go really into details about the genes or ST cal types. I just want to give you an overview about these combinations and how effective they are compared to each other. So first, about Klebsiella pneumoniae. In this figure, you can see on the x-axis the antibiotics and combinations listed, and on the y-axis there's the percentage of resistant bacteria. The reason I added the antibiotics alone as well is that I measure the MIC levels of the strains against the antibiotics alone as well to compare them to the results of the combinations. And for example, in the case of Astronam, there was almost 90% of the bacteria were resistant. So let's move on to the maybe most eye-catching result here in this figure. In the case of Astronam avibactam and Astronam nacubactam, all the strains I had were killed by these combinations, meaning that they are really promising combinations for the future. I would point out here that the rules are of drug prescription now is that if a clinician has a patient that can be saved by any other drug that is available that completed the trials, then they are able to pick out a drug that is in a phase 3 clinical phase to save the patient. And Astronam avibactam has been actually used before such way that the clinician gave this combination to a patient and saved its life, because no other antibiotic, no other drug was actually effective against that infection. Then about cefepime and its combinations. You can see that cefepime nacubactam and cefepime taniborbactam were quite promising, and they had a low resistant rate. I would also want to talk about cefepime N-metazobactam and cefepime tazobactam. You can see that they have almost the same level of resistance, which is interesting because cefepime tazobactam is an older combination, and N-metazobactam is a new one. But the two inhibitors, enmetazobactam and tazobactam, are really well close to each other in a case of chemical structure, and that's why they don't really have differences between the two resistance rates. So we can say here that all the money, time, and effort that were put into developing cefepime and metazobactam weren't quite effective. We could have used it better ways. And I also pointed out the combinations of nacubactam, because as you see, we combined it with 3 different antibiotics as well, and all of them were quite effective, so they are all quite promising. So moving on to Acinetobacter baumannii, I've got fewer combinations against Acinetobacter. It's also because of the not so many combinations are being developed against this species. But here I also got one combination that was completely effective. So Bactam durlobactam, it also killed all of the strains I had against this combination. So it's as well quite promising. So to conclude everything I was talking about, I tested 230 strains against 13 different combinations in order to see whether these combinations are effective, whether there is actually a resistance to them right now, even that we didn't start to use them in the clinics and in the hospitals. It points out that these genes can be transferred, and there are a lot of resistance genes in our environment that are actually really dangerous, and they can provide resistance to even these newly developed drug molecules. And after we are going to go more into details about the genes, about all the strains, we are going to have a bigger picture about the effectiveness of these combinations. And hereby, I want to mention my mentor, Bálint Kincses. I'm really grateful for all of his help and advices during my work in his lab. And I'm also really grateful for the National Academy of Scientist Education, because they are the ones that are responsible for me being able to be part of this lab, so I'm quite grateful. And also, thank you for your kind attention. If you have any questions according to the presentation, I'm going to be around, so please reach out to me and we can talk about it. So thank you very much.
Thank you for the nice presentation, and now please take your certificate.